we are going to continue the soil miracles from a scientific perspective with soil biologist Erik Joner that you know from earlier today. I welcome Erik up on stage.
Tack. Yes, thank you again. This is short soil science 101 with a focus on the extraordinary. Yeah, as I was introduced, I'm a soil scientist, agronomist from this university, having worked for, yeah, what will it be now? Some 40 years on soil soon. As a mercenary of soil science, in research you have to search for contracts on different topics that are fashionable and important to society. So I've been shifting between classical soil microbiology, which I did my master's and my PhD in, working with mycorrhizal fungi as a working horse, later applying this to soil pollutants, organic pollutants, heavy metals, radionuclides, and studying soil pollution in many ways, now most recently on microplastics and antimicrobial resistance. And in the meantime, also working on soil carbon sequestration, biochar, green roofs, urban agriculture. So I'm a sort of a jack of all trade on soil science, and I will talk about some of the fundamentals of soil science which apply to all these areas, and that are key to understand how both organisms and nutrients and pollutants move and exist in soil, and how they are transferred to food and to organisms, to plants, etc. So the challenge was to make something a little bit different from the usual, talking then about the soil miracles. First, a little introduction to the importance of soil. The soil is the best basis of all terrestrial life. There is, of course, life in the sea, which is not so much affected by soils, but all the plants we see, all the animals feeding on plants, and the rest of the food chain feeding on them again, they depend on soil. Of course, the ultimate source of energy and carbon is the sun and the CO2 in the atmosphere. But with this as an energy input, soil is creating an enormous diversity of niches where different plants and different organisms can thrive. And this is the basis then for almost all terrestrial life. Another very important thing that soil does is that it degrades and recycles all elements, all mineral nutrients, and they degrade all organic compounds so that they enter back into the elemental cycles where they can be reused again by new plants or by different animals. And water issues with soil are extremely important, both to plants and also to us as humans. And the fact that soil is a porous medium that is retaining soil so that water doesn't come into the streams and rivers as soon as it rains, that's an extremely important buffer in the world and terrestrial world. And through this process, soil is also purifying water so that what comes into the groundwater, into the streams, are usually cleaned from dissolved organic matter, from pollutants, from organisms, and so on. So this function of soil as a filter is also crucial to life on Earth. Then soil is buffering climate change. Soil is a huge carbon storage pool. But it's also, of course, buffering temperature fluctuations because it contains water and buffers temperature changes in forest and other vegetated systems. Pollutants or toxins are either immobilized, as for the heavy metals and radionuclides and so on, in soil, or they can be degraded or immobilized if they are organic. These are natural organic molecules that are harmful to us and to plants and to animals, or it can be man-made pollutants like pesticides or all the chemicals that we use in consumer products. This is a very important thing about soil, that it sort of immobilizes, sequesters, degrades, and doesn't transfer on these pollutants to living organisms. And then soil is an enormous biodiversity pool. You've surely heard that a teaspoon of soil can contain 10,000 species. And this is beyond imagination and becomes surrealistic in some way, but this is far beyond any other ecosystem regarding biodiversity. Then, of course, these organisms also represent a treasure chest for biomolecules. We know that a lot of the antibiotics made by the Streptomyces family are saving lives every day as antibiotics for human health and veterinary medicine. Unfortunately, they are also seeping into the environment in quantities that we dislike and affecting organisms regarding antimicrobial resistance. But nonetheless, it's a pool of biomolecules that we can exploit. Soil is also a cultural archive. All archaeological excavations are made in soil. Soil can preserve things pretty well, or it can degrade it and make it disappear. So sometimes we find things that are interesting in soil from way back, other times it's gone. And then finally, soil is a very good lesson, or a good sandbox for learning lessons on ecology, on art, on you name it, chemistry, physics, botany, a lot of things thrive in soil. So these are what we call ecosystem services, and I don't like ecosystem services as a term because it's putting value on soil, monetary value, and I think, or it's putting a price on soil in many contexts, and the price of soil and the value of soil is often very different. If you build a road, a farmer will get some 30,000 kroners per dekar, or for 1,000 square meters of soil, which is then taken out of production entirely, and this is supposed to cover his future income from that soil forever, which is briefly, or which is for practical reason, shortened to 30 years. So that's the value of soil to an economist. To us that know that soil is irreplaceable, the value is far higher, of course. So if you like ecosystem services as a way of transforming value into money, then you can embrace it, or otherwise be a bit cautious. One of the first miracles of soil is its porosity, and then we're first talking about the mineral basis of soils, because there are all the biological miracles as well. But to understand the porosity of soil is understanding soil. This porosity is crucial for the soil functioning. The pore space in soil is roughly constituting half the volume. If you have a liter of soil, you actually have half a liter of void, of holes, and half a liter of solid material. And these pore spaces, these empty voids in the soil, vary in size, and they are filled with air or water. And in the water there are dissolved nutrients and biomolecules of all sorts. Everything on communication and metabolism happens in water, not necessarily floating water, but on the water films covering surface of particles. So in an ideal soil, the pore space is half of the volume, and half of this pore volume is filled with air and the other half with water. Then there is enough water for metabolic processes, and there is enough air for everyone to breathe there. Most organisms need oxygen in soil, including plant roots, of course, which die if there is no oxygen. I said the nutrients and all the biomolecules are dissolved in water found on the films covering the pore surfaces. And this is why pore surfaces are important and a very highly overseen property of soil. So all the plant nutrients are, for example, found there, and all the dissolved organic molecules that bacteria and fungi and other organisms live from. They are not floating around, or they are not floating in the air of the soil. They're in the water of the soil, and to some extent they're of course also bound to the surface itself, not dissolved in this water film, but only accessible through this water film. So the surface area of pore walls are mind-blowing. If you thought the 10,000 species of microorganisms in a gram of soil was a lot, then think of the pore space volume and the specific surface area, as we call it, the number of square meters per gram of soil that you find of surfaces within a soil, because it's a question of square meters. Even if a gram of soil can fit into a cubic centimeter like a Yahtzee die. A teaspoon of soil thus has more surface area than an average apartment. So if you live in a 50 square meter apartment, this is as much floor surface as you can find pore space surface in 1 gram of soil. This is because clay constitutes part of the soil, and clay particles have a very high specific surface area. Depending on the clay type, it has around 50 to 800 square meters per gram, which is quite surrealistic. A gram of humus, or transformed organic material, stable organic material, has even more, between 800 and 1200 square meters per gram. And of course, soil doesn't consist of only clay and humus, but if an average soil around here would have 20% clay and some 3%, 4% organic matter, this this becomes a surface area which is equivalent to the floor area of a 2-room, 3-room apartment. So that's where plant roots need to find their nutrients. And since bacteria don't swim around in soil, but they are stuck on surfaces, means that they're not exchanging very much unless they can give signals through the water film, which is extremely tortuous. Tortuosity, it's a weird word, but it means the sort of detour that you have to make from one place to another if you have to pass through a meandering river or a pore system within a soil. This is also why organisms are in soil can be so diverse, because they don't interact. They are often closed in to small pores where they don't communicate with other organisms that can be just micrometers away. Turning to biodiversity and biological activity, that's maybe where the real miracles, or at least the more colorful and interesting part of soil is found. It's true, soil is teeming with life, but in fact most soil organisms are inactive most of the time. They are dormant, and that's maybe a good thing, because if they were active, they would do what they do best. They would degrade organic matter, and most of the stable carbon in soil would be in the atmosphere. Also, those that are active, they are starving most of the time. So soil is a dark, cold, sad place, actually, which is, of course, unfortunate when you try to communicate the joy of the marvels of soil. But soil is a really shitty place to be. Then again, what do all these organisms do? The diversity is because there is different niches and different life strategies among soil organisms, yet more than 99% of the organisms living in soil do the same thing. They degrade organic matter. But they have different preferences and different affinities for different type of organic matter, and they are found in different places. They have different growth preferences when it comes to pH or temperature or oxygen, partial pressure and things like that, which gives them unique properties during certain times or in certain niches. So, in spite of all these limitations, the biological miracles abound in soil. Maybe the most important one is the nitrogen-fixing symbiosis. Most terrestrial nitrogen comes from symbiotic bacteria living off photosynthesis products from plants. The legumes is the most important family, maybe, but we also have trees that form different type of root symbiosis, and what happens is that these plants develop small nodules on the roots where the bacteria is proliferating and have a direct food source from the plant. The plant can produce sugars from photosynthesis in large amount, but it can't grow unless it has mineral nutrients. And the main mineral nutrient limiting plant growth is nitrogen. So the plants that can have their own nitrogen source through bacteria, where the bacteria takes the nitrogen from the air. Air is 78% N2, which is not useful to any organism except some very few bacteria that can transform N2 into ammonia and make it bioavailable. And this is usually a process happening or nowadays this is a process that's happening industrially, and it takes huge amounts of energy and hydrogen and so on. These plants can do this at low temperature, low pressure, and for free. And still, I will maybe go a bit further here on my keywords. This is a free and widespread type of symbiosis, which provides still a lot of the nitrogen that agriculture is based on today. The secret here is that these nodules contain a type of hemoglobin, so it's an oxygen sequestering molecule, because these bacteria, they have an enzyme which is extremely oxygen sensitive. If there is oxygen there, they can't fix nitrogen. So what the plant does is to provide a microenvironment for the bacteria where there is enough nutrients, sugar, and a high respiration rate and a high oxygen sequestering mechanism, rendering the inside of the nodule anaerobic, permitting this nitrogen fixation process. As I said, today, still about 50% of what is taken up of nitrogen of plants throughout the world comes from this symbiosis, and the other half is based on the chemical nitrogen fixation. Not so much Birkeland process anymore because it's industrially much more expensive and energy demanding, but on the Haber-Bosch process, which, contrary to the plants, require 500 degrees, 200 bars of pressure, and a massive input of hydrogen, which is a very energy-rich gas having to be produced from methane or from natural gas, fossil gas. So this is a free miracle to be used in agriculture and in all sorts of soil improvement schemes. It's also key to sequester more carbon in soil, because soils as a carbon pool, if we're going to sequester carbon by enhancing soil organic matter, we have to be aware that stable soil organic matter has a C to N ratio of 10. So it means that for every ton of carbon we bind or sequester in soil, we need to sequester 100 kilos of nitrogen. And that's in competition with plant nutrition, because this nitrogen can't be both a plant nutrient and a stable carbon pool in soil. You can't sell the butter and have both the butter and the money for the butter. You need to choose. So either it's the nitrogen going to feed plants for their growth, or it's nitrogen that is going to be sequestered with the carbon. And the second miracle of biological miracle of soils is called Mycorrhiza. It's a fungus root symbiosis acting where the fungus is acting as the roots of the roots. If you imagine a cubic centimeter of soil with 50 square meters of surfaces inside it, and the amount of roots that can be found in a cubic centimeter of soil, you're usually limited to about 2 to 3, 5 centimeters of roots. And if you're in a 50 square meter flat and you're just 5 centimeters long, you don't go very far to try to find the nutrients that are scattered all over that floor. If you have the help of a mycorrhizal fungus, you will have an extra 20 meters at least of absorbing organs, and this is then helping the plants exploit these 50 square meters of soil per gram of pore wall surface. So the main mechanism of this organism is to scavenge soil for nutrients and transport them back to the plants. It's actually not producing new nutrients, it's just making sure that the plant get hold of the nutrients that are already in the soil. But the mycorrhizal fungi also do other things. They can enhance plant health, they can provide a network of subsurface exchange, signaling between plants, signaling between fungi. These fungi can be linked to different plants, both within the same plant species, or between different plant species, and in some situations, for example, if you're in the forest and you cut down a big tree, and there's a smaller tree hooked on to the same mycorrhizal network, this small plant will take over the whole fungal network for its nutrient supply. To start with, the big tree is actually the one benefiting from being hooked on to this network, because there is a source-sink relationship in this in the benefit of mycorrhiza. The one that provides most sugar through photosynthesis gets the most mineral nutrients back. But when the tree is cut down or dying from other reasons, there are other plants hooked onto this system, and then they will start to benefit. That's at least the main quantitative benefit of this for forest systems, seen in a sort of a productivity way. Then I'm a little bit reductionistic here, but that's the scientist in me. Mycorrhizal fungi are also building soil structure. It means it's helping to form aggregates so that soil is stable when it comes to erosion. It's making a network of hyphae that is connecting particles or lumps of particles into a more sturdy and erosion-resistant matrix. So, for those of you who are not familiar with the mycorrhizal fungi, I don't know if my pointer is showing here, is it? No. Whoops, I moved on here, it seems. Well, anyway, what you see to the top, on the top here is what is called arbuscular mycorrhiza, which is colonizing herbaceous plants, while in forest systems you have something called ectomycorrhiza, colonizing forest trees and also forming these toadstools or edible mushrooms that we collect in forests. Those are very different fungi that do different things. So sometimes you hear fabulous stories about mycorrhizas that can do this and that. Sometimes it doesn't apply to all systems. So be aware when you hear stories like that there are some differentiation between these two. These are two of the main groups of mycorrhizal fungi. There is another very important one, which is colonizing heather species like blueberries and heathers in very high altitude, high latitude, cold acidic soils. Then there is a compartment of soil called the rhizosphere. It's the zone of soil surrounding roots or in immediate vicinity of the soil of the root surface, and the rhizosphere is a hotspot in soil. When I said that most organisms in soil are dormant, they're a little bit like the Sleeping Beauty. The rhizosphere brings in nutrients, a trigger for activity. It produces a liquid called root exudates, which contains sugars and a lot of other nutrients, which boost microbial activity. And this is the prince then bringing all these dormant microorganisms to activity. So this is one of the major hotspots for soil microorganisms in soil around root surfaces. But root surfaces isn't a single thing. A root grows on, and the rhizosphere is actually only active close behind the root tip, and as a root grows on, the older part of the root will be subrinized. It will be dark and corky, and it will not exude these juicy root exudates any longer. So rhizosphere is not only a place in soil, it's also a time in soil. So some people seem to think that the root exudates are everywhere all the time. They are not. They are only around the root tips for a brief period. And the roots don't live very long either. Some roots are only live for a few weeks. Some live for years, but that's the one you stumble over when you walk in the forest. Most small active roots have a very short lifetime. Yeah, so this brings dormant bacteria into life, and these bacteria, they help the plant mobilizing both nutrients and other things in soil to improve plant nutrition. Some of the root exudates in themselves are acids and mobilize mineral nutrients that are bound to surfaces, which are detached by these acids. But sometimes also the bacteria produce acids or other type of molecules that are mobilizing plant nutrients. And then this is it's not a true symbiosis. It's more like a functional two-way exchange. But both organisms can live perfectly well without the partner. So that way it's not what they call mutualistic symbiosis. Then the root exudates that enter into the soil is also a carbon source for soil, not only for the soil organisms, but also for the soil itself. Partially this is because these rhizosphere organisms, they bloom for a period and then they die. And this necromass, the dead soil organisms, are part of or will become part of the stable organic matter in soil and constitute part of the carbon pool of soils. Worms and other soil engineers. There are plenty of larger animals than microorganisms. The macroorganisms are mainly doing the important work of shredding organic litter into smaller pieces that are more accessible to smaller soil animals. Of course, there are a gradient of sizes from the bacteria towards passing through the protozoa, for example, which are bacterial grazers, and which is also one of the reasons why bacteria don't accumulate in soil. They are eaten by these protozoa, and their nutrients are released. Their recalcitrant cell walls are expelled again, and you have the bacterial necromass. Then again, the protozoa are fed upon by nematodes or other small soil organisms that again mineralize part of the carbon and release mineral nutrients. But for all of these smaller organisms to be able to feed on litter or manure or other things that we put into soil, we need the shredders and the mixers, and those are the larger soil animals like the earthworms, the potworms, the mites, the insects, the small crustaceans that you recognize on the bottom left. And this mixing and shredding is boosting turnover of both carbon and nutrients. The intestines of these organisms is the second biggest hotspot in soil for microbial activity. So when bacteria lie dormant in soil, they can experience, or they are likely to experience, two things that will boost their activity. One thing is that there is a root growing past it, and there is carbon source from the root exudates giving them abundant food. The other possibility is they are eaten by a worm or an insect or something, which puts it into the intestinal tract of this macrofauna organism and mixes it thoroughly together with mineral particles and other things that are accidentally ingested together with the organic material that the organism wants to digest and obtain energy from. So these intestines is a very important hotspot for microbial activity. And as for a rhizosphere, it's a very temporally short happiness. You get eaten, you have a party for a day or so, and you get shitten out again, and then you wait another year until you're eaten again. That's basically the life of a soil bacterium. Soil worms also bury organic matter and bringing it down from the surface to deeper layers, which is important both for improving soil quality at depth, but also, for example, for archaeologists. If earthworms leave their casts on the surface, Charles Darwin actually measured that as much as 5 millimeters of soil is brought from deeper grounds up to the surface, and burying things like ruins from ancient civilizations. So archaeologists should be very grateful of earthworms, because they have ensured that archaeological dig-outs can be made, and that the things that they find pretty deep in the soil have been buried in soil and not eroded on the surface. Charles Darwin published this book on earthworms a year before he died. It's freely available on the internet, so you can find it and read it. And it's pretty funny because it is experimenting in a very classical, funny way, trying to deduct if earthworms are intelligent. Are you intelligent if you can pull down a pine needle by the bottom of the V rather than the top? You can't get a V-shaped needle into the soil if you grab it there, because the other part will stick. But if you grab it by the bottom, you can pull it down. Earthworms manage that, and Darwin found out that even if you clip off the stinging point of the needles, they can figure it out. So he claimed they were intelligent. I'm not quite sure, but they at least have some good instincts. So. The final and maybe most important things that the earthworms do is also that they make channels in the soil that are good for drainage of water and aeration of soil. And these earthworm burials are also used for, or these earthworm channels made by the anesic earthworms. There are 3 groups of earthworms. The anesic ones are the one making permanent channels in soil, and those channels are also occupied by plant roots. So plant roots can go very deep if they find earthworm burial channel and following it down to the bottom. Yeah, finally, soil or earthworms are also used as soil health indicators. So if they are plentiful and active, it's usually a healthy soil. If they have a thermometer in their mouth, it's my daughter who drew these. They probably don't live in a very healthy soil. Last slide on soil biodiversity. Soil biodiversity is unfortunately a rather ill-defined parameter. I said there were 10,000 species of bacteria in a gram of soil. Is a soil with only 5,000 a much worse soil? It's a pertinent question of redundancy, and it's also a question of what does a new species bring? Isn't it an introduced species? Does it have anything to do there? Do we extend the species concept to new genes? A bacterium with a virus coding for antimicrobial resistance is maybe not a new species, but there is a new gene in there, and you can say that this is higher biodiversity when it's introduced. But it's not necessarily something we would think of as beneficial to a soil. Yeah, this species richness is also a bit of a tricky metric because it doesn't take into account function. Are another 1,000 new species that do almost the same thing as those that are already present do anything good to soil, or is it just increased competition and impossibility for tilting an equilibrium that's already there? And how about the other metrics for soil biological interactions? As a microbiologist, we are much more used to looking at activity. How much activity is there in soil from organisms? How much biomass is there? How many are they? How big are they? And maybe more importantly, how are the organisms in soil doing what they're supposed to do? Are they emitting methane or nitrous oxide or other greenhouse gases that we don't want them to release? Or are they protecting plants against diseases? 2 very different functions, and whether there is many or a few species doing this is more interesting than if there are many species per se in soil. And especially for some soil functions that depend on very few different species. The mycorrhizal fungi that I told you about, there are only around 200, 250 species worldwide of these arbuscular mycorrhizal fungi, which are crucial for plant phosphorus nutrition. They might be surveyed when it comes to biodiversity and abundance, but for organic matter degraders, if there are 10,000, 8,000, 5,000, wouldn't really change the function of soils. And then biodiversity is also a little bit of a tricky thing. We know a lot about biodiversity above soil, but the part of biodiversity that we are considering is in the little white box to the bottom right there. There are enormous amounts of biodiversity that we don't know much about, belonging to the archaea and the protobacteria. So, and fungi also, of course. So, well, biodiversity is not always the metric to examine in soil. Yes, That was a leftover from the last slide. Don't think this plant carbon improving soil belonged here. Well, that was what I wanted to learn you on soil miracles and crash course of soil science, which usually takes half a year. Sorry if I went a few minutes over time.
Thank you so much, Erik. We have time for one short question before we have a short break with some sweets and coffee and tea. Does anybody have a short question for Erik?
There are no short questions on soil. So.
No, maybe.
Maybe it's not a short answer, but I'm curious about soil health because it has caught on in certain contexts as, yes, soil health really captures something important. But I also know that from a scientific perspective, soil health is not really useful because it's ill-defined or difficult to define. How do you approach soil health?
Yeah, I don't like the term because it's not very well defined. But soil health is a good way to attract attention to a serious problem about soils. That's where it has its merit. The metrics that they want to measure to determine if a soil is healthy or not is unfortunately imperfect. I mean, the only difference between soil health and soil quality is the biological part. And the biological part is unfortunately then allocated to biodiversity rather than function. But I go along with soil health because it has a good purpose. But as a microbiologist, I dread having to determine if a soil is healthy or not. Using microbiological tools. You can always look at erosion or on other aspects which is related to soil health and say, okay, this problem has a problem, this soil has a problem, or it's not scoring bad on this soil health parameter. Yep.
And coffee, and then we're going to continue again after that.
Yes, you can.
So we're going to have a few minutes where you can walk down and have some sweets and coffee, and then we're going to continue again.